So we've been using these rapid antigen tests a lot lately to see if we have COVID-19. But how do they actually work? So if my snot has coronavirus in it, then when I apply this solution to the test, it should register with 2 lines at the C and the T. If I don't have a corona, and if I have a negative result, then I should only have a line at the C line. Sweet, I'm negative. Also, what just happened inside of this little magical box? Well, let's open it up here and see if we can find out. Aha! I knew it! There's a magical box on the inside that makes it all work. I knew that mysterious box was really weird. Okay, not really, but if we want to understand how the test works, we have to understand the structure of the COVID-19 virus. Okay, here I drew a picture of the virus, and what we really need to understand is this spike protein right here, okay? It's covered in these spike proteins that make up the capsid of this virus. Here's an even more in-depth image of that spike protein on the coronavirus, and here's actually a 3D model of that protein. So a protein is really just a complex polymer macromolecule that is made out of a bunch of smaller molecules called amino acids. And you can see each of those individual amino acids highlighted pink right here. In fact, if I just click on this and zoom in, that right there is that's a tyrosine amino acid. That right there is an isoleucine amino acid. That's a glycine amino acid. That's a lysine. And I can click through this whole model. This is an isoleucine, and each of these little segments, that's an aspartic acid, of this protein on the coronavirus is made out of these small little amino acids. That's really important because when I mutate those amino acids and I change them around, that's when we get variants. Here's another model of that spike protein, and this little amino acid right here and this little amino acid, the ones in red, they like to mutate a lot. Every time that there are significant mutations in the structure of the spike protein, that's when we get a variant of the virus. So for instance, the Delta variant is when we had those mutations in those amino acids right there. And right now, at the time of this video, we're dealing with the Omicron variant, which is these particular amino acids have been mutated inside of the structure. So it's a slightly different structure. In fact, it's very mutated compared to the other variants. So what actually is the purpose of this spike protein? The spike protein is basically a key, and it helps unlock your cells so that it can get into your cells and then attack it. And this spike protein, or the key, is what we call an antigen. To help me remember that, I kind of think of a lady named Jen.
Hi!
Okay, not that Jen. Actually, we're gonna spell her name with a G. Okay, so the coronavirus is antigen. The antigen wants to attack Jen, and this is a big problem for Jen. Jen's body is actually just freaking out right now, and the alarm is going off in her immune system because the antigens are attacking. Ah! What is Jen going to do? Well, I'll tell you what she's gonna do. She's gonna make antibodies. Jen's antibodies are going to attack the spike protein's antigens so that it can protect her from the virus, okay? So actually she can be happy now. That's interesting. So these antibodies can stick to the spike proteins of the coronavirus. That could be really useful. So maybe if we take these sticky little antibodies and we put them inside of paper and then stick that paper inside of a magic little box, then we can actually see if they stick to any coronaviruses inside of our system, and then that tells us if we have the virus or not. That's a great idea. Let's invent it and call it a rapid antigen test. That's cool and all that, but how will we actually see the antibodies that attack the antigens if they're so tiny and molecular? That's a good point. I know, let's put gold in it. Gold. When you take really small nanoparticles of gold, they're actually colored red, and you can use them to color code the proteins or the antibodies that we're going to use in our antigen test. The antibody proteins are Y-shaped. They kind of look like somebody standing with their arms raised up, like they're doing the Y in the YMCA. And their hands are out, and they're specifically designed to attach to the amino acids on the coronavirus spike protein. So they're very useful for this test. The antibodies are so small, though, that the only way we can see them is if we tag them with gold. So we take those gold nanoparticles and attach them to the antibodies. All right, we're gonna imagine that this piece of paper is our antigen test. So this side of the paper is going to represent where we put our snot into. So the people who made this little rapid test put those gold-colored antibodies inside of that well that you drop your snot into. Then they created a test line with antibodies that aren't gold-colored. All right, I've got my antigen test set up. Let's say that I give it some coronavirus from my snot. All right, so there it is inside of the beaker, and I'm gonna pour it onto this antigen test. If the solution you put into the test has the coronavirus in it, then those antibodies are going to stick to those viruses trying to attack them. The solution then moves up the paper, carrying the antibodies with them, and they meet the new antibodies at the test strip. Those antibodies also want to stick to the virus, and so they do, which means that they also attach to the antibodies that glow red. Further up the test, you've got antibodies that are solely attracted to the antibodies themselves. So these act as the control line. It just shows you that the test is working and that it actually has antibodies that glow red in it. So this morning when I took my negative test, that control band right there is a band of gold particles that are attached to just the normal antibodies. The reason I have the control strip is it's telling me that the strip is it's working like it should. There are antibodies present and the antibodies have gold on them. The only reason a test strip should show up with a red line is if there are viruses that the antibodies have attached to. This is actually an experimental setup inside of this little cassette right here. So the control group and the experimental group are the two main groups that you would include in any experiment with the scientific method. The independent variable that I'm introducing here is the coronavirus, and the dependent variable is whether or not two lines show up. So my hypothesis would be, if I have coronavirus, then two lines would show up, and I have these two groups to compare the results, and the second group should only show a result if my hypothesis is true. So antibodies are a type of protein, and they're made of amino acids, and the coronavirus is made of a protein capsid, and those are made of amino acids. And all the variants that you hear about in the news, like Omicron and Delta, those are all caused by just changes in those little amino acids. So if we can understand how to build a protein, then we can also understand how to build an antigen test. So understanding how proteins are built seems really important, which is what we're going to study in Unit 5. So here's a question. Where did all of these coronaviruses that are in your snot come from? Who built them? Well, you did, because the virus is hacking your system. All right, here's the virus again, and all of these proteins in this entire system here is being made by your own cells, which are producing them using the ribosome, okay? You'll remember, if the mitochondria is the powerhouse of the cell, the ribosome is the protein builder of the cell. So where does the ribosome get the instructions for how to make the viruses in your cells? That comes from the nucleic acids inside of the coronavirus, okay? That's this helix that you see right here, okay? It's either DNA or RNA that is inside of the viruses, and the coronavirus has RNA inside of it, okay? So nucleic acids like RNA or DNA are the instruction manual for building the proteins. So if we can read the DNA or the RNA code just like the ribosome does, then we know and understand how to make proteins. And that's exactly what we're going to learn in this unit. So when you get infected with the coronavirus, it forces your ribosome to read this code and then to make more of the viruses. And how does the ribosome know how to read this code? Well, it uses a key to decode it, and that key looks like this. Now this may look super complicated, but you're going to learn how to read this in this unit. So go ahead and start on your notes.